Good morning, students. So in the last class, we discussed regarding about the body plan as well as the body wall. Now today we will discuss regarding about the body symmetry as well as the metabolism and cephalization. So let us start the one by one. The body symmetry. What do you mean by the symmetry? So as you know that the most of the organisms are most obvious feature of the animals is the presence of the general body axis. The presence of the general body axis. That is central axis of the body. Central axis of the body. So this central axis of the body is mainly formed by the aggregation of the various organs as well as organ systems, or maybe the various tissues around the central axis of the body. Around the central axis of the body, the various organs as well as organ systems are gets arranged in a systematic pattern that helps in formation of the central axis. So this is central axis. So when individual possessing this central axis or maybe general body axis, so when we cut the individual's body either in a single plane or multiple planes, either in single plane or the multiple plane, then we will get the mirror image halves. Mirror image halves. So that is called as symmetry. That is called as symmetry. For example, if you see the various organisms, like right from the Polyphenols up to the higher animals like the mammals, so the most of the animals, they are exhibiting the general body plan. They are possessing the general body plan. They are possessing the central axis of the body. So where we are differentiating into the body, the anterior as well as posterior surface, dorsal as well as ventral surface. So towards one end there is a mouth. This mouth forms the central axis of the body. Otherwise, the one of the opening present towards the one end is forms one of the central axis of the body or the general body axis. If you cut the body through this central axis, in a one plane, in either this plane or this plane, or in any plane, if you cut the body through the central axis in a single plane or more than two planes, then we will get the mirror image halves. Where we are going to get the mirror image halves. We are going to get the two equal body parts. So that is called as the symmetry. So as you know that some of the organisms belongs to the certain groups or maybe certain phyla, they are exhibiting asymmetry. This asymmetry is mainly because of its shape. The body shape of these organisms. The shape of the body organisms is somewhat different. It is irregular. There is no symmetry. There is no definite shape. In such condition, the individuals are going to be exhibit the asymmetry. So here the symmetry is the one of the main basis of the classification of animals. Based on that, we are classifying the animals into the different groups. So which of those symmetry are? Let us study one by one. The first one is asymmetry. The individuals they are exhibiting the asymmetry or maybe irregular body shape. They do not possessing any general body axis, any general body plan. They do not possessing any general body plan or body axis or the central axis of the body. Here there is no definite shape. So these individuals they do not have definite shape. The body shape is not definite. It is irregular shape. In such condition, the general body plan we cannot observe. There is the absence of general body plan. In such type of individuals, if the cut body in many plane, then we can't get any the equal halves or maybe the mirror image of halves. So, say for example, here it is the Polyphenols. The Polyphenols like the sponges or spongilla. The sponges they are possessing the irregular body shape. Otherwise, if you observe the corals, otherwise if you observe the snail belongs to the phylum Mollusca. Corals belongs to the phylum Porifera. Sponges. So these individuals are belongs to the Poriferans. Certain individuals they are belongs to the Coelentrates also, and these individuals they are not exhibiting the symmetry. That is general body plan. They do not have any general body plan. Then central axis is absent. If the central axis is, if it is absent, if there is no shape, definite shape of the body, in such individuals they are exhibiting the asymmetry body plan, body symmetry. That is, for example, best example if I take the example is here it is amoeba. If you take the amoeba, the body shape is irregular. This is the amoeba where the body shape is irregular. If you cut the body in this plane or maybe this plane or maybe this plane, we can't get equal halves of the body parts. We are unable to get the mirror image halves. Mirror image halves. So that's why such type of symmetry is referred as asymmetry. The asymmetry is common as in case of sponges, in corals. Even though in snails, the snails, although they are belongs to the molluscans, the molluscan individuals they are exhibiting the bilateral symmetry. But one of the member of this phylum called the snails, they are exhibiting asymmetry. This is because of there is no general body plan in this individual. So that's why. So this is the one of the symmetry is observed in some group of organisms like the Poriferans, as well as molluscans and some other organisms. So that is asymmetric. Next one is Radial symmetry. So here here there is the arrangement of particular organs or maybe the tissues or organ systems around the general body plan, around the central axis of the body. Around the central axis of the body, there is a definite organs as well as organ systems, or might be the tissues are aggregated that helps in formation of the general body axis or maybe the central axis of the body. The central axis of the body, if you cut the body of this individual passing through the central axis of the body in single plane or in many plane, we are going to get the equal halves. But as in case of radial symmetry, so if you observe example for radial symmetry is the individuals belongs to the coelenterate. The individuals belongs to the ctenophora. Some of the ctenophorans they are exhibiting the radial symmetry, but some of them are exhibiting the biradial symmetry. But here, the most of the ctenophorans they are exhibiting the radial symmetry. Say for an example, the sea anemone. The sea anemone. Otherwise, if you observe the Aurelia, Obelia, Hydra, so these individuals belong to the coelentata, they are exhibiting the radial symmetry. If you observe this individual belongs to the coelentata, if you cut the body of this individual through the central axis, this forms the central axis. Because this individual mainly possessing the blind sac body plan. The blind sac body plan individuals possessing a single opening towards the anterior end of the body. Through this single opening only, they are involving in ejection as well as ingestion process. So here is the single opening is there. Through this opening only, both the processes takes place. So this single opening is forms the central axis of the body or the general body plan. Passing through this single axis, if you cut this organism, not in single plane, more than two planes. If you cut this organism, so this is the central axis of the body. If you cut this organism through the central axis, in this plane, we are going to get the two equal halves or the two mirror images. Otherwise, through this axis, otherwise through this axis, in this plane, or in this plane, or in this plane, or in this plane. Here is, we are going to cut the body of organism in more than two planes, not in single plane, more than two planes, passing through the central axis of the body, passing through the general body plan. So where we are going to get the two equal halves, the mirror image halves, or two equal body parts. That is called the radial symmetry. That is called the radial symmetry. So here, the definition is when we cut the body of an organism more than through the central axis of the body, in more than two planes is called as the radial symmetry. So when we cut the body of an organism through the central axis of the body in more than two planes or more than three planes, then it is converted into the two equal halves. That is called as the radial symmetry. So this is more commonly observed as in case of the coelentrates. The individuals belongs to the coelentrata and the individuals belongs to the ctenophorans. But some ctenophorans they are exhibiting the radial symmetry, but some of they are exhibiting the biradial symmetry. At the same time, the adult echinodermate individuals. Adult echinodermates. The adult echinodermates, they are also exhibiting radial symmetry, but not the larva ones. The larva of echinodermates, they are not exhibiting the radial symmetry. They are exhibiting the bilateral symmetry. So that is the example of the radial symmetry. Then the bilateral symmetry. So the bilateral symmetry, the name itself explains here. The bi means the two lateral parts of the body. The two lateral parts of the body, they are helps in formation of the two equal halves. When we cut the organism by passing through the central axis in a single plane, not in more than two planes, not in two planes, only in single plane. When we cut the body of an organism in a single plane through the central axis, then it is gives us to the two mirror image halves. That is called the bilateral symmetry. The body of an individual cut a single plane passing through the central axis of the body, then we will get mirror image hands. That is called bilateral symmetry. So the body of an individual cut in a single plane, only in single plane. So here in radial symmetry it is multiple plane, more than two planes, passing through the central axis of the body, passing through the central axis of the body. Then we will get the mirror image hands. That is called the bilateral symmetry. If you take the higher animals, right from the platyhelminthes or the right from the annelidans, don't take a platyhelminthes, right from the annelida, arthropoda, molluscans, chordates, all the chordate individuals, all the chordates, except a few individuals, few organisms, they are exhibiting, the most of they are exhibiting the bilateral symmetry. Most of the animals, they are exhibiting the bilateral symmetry. Because in this group of organisms, where the different organs as well as organ system, they are gets arranged around the central axis of the body in a coordinate manner, and that helps in formation of the two equal halves, that is two mirror image halves when we cut the body through the central axis in a single plane. That is called bilateral symmetry. So here in this the most of the organisms, the molluscans, arthropods, annelidans, they are exhibiting the bilateral symmetry. They are exhibiting bilateral symmetry. Say for example, if you take the human beings, if you take the humans, in human body, so this the central axis is mainly formed by the sternum. Here one bone is there towards the ventral surface of the body in human beings. Here a single bone is there. This single bone is called as sternum. So this sternum is forms the central axis of the body. If you cut the body of human beings in a single plane, only in this plane, not this plane, not this, only in this plane. In this plane, if you cut the body in a single plane through the central axis, it interfuses to two equal halves. One side, one eye, one ear, half portions of the teeth, half portion of the tongue, one arm, one leg. That is the two halves which are produced when we cut the body in a single plane passing through the central axis. Then each and every part of the body, lateral part of the body, they are possessing the same structures. They are possessing the same structures. That is called as a bilateral symmetry. The most of the organisms, they are exhibiting the bilateral symmetry. Next, biradial symmetry. What do you mean by this biradial symmetry? So the biradial symmetry is exhibited by the organisms belongs to the phylum Echinodermata. The organisms belongs to the Echinodermata. So the bilateral symmetry in the sense during larval stage, organisms exhibit bilateral symmetry while in adult stage they exhibit radial symmetry. That is called biradial symmetry. During larval stage of an organism, of an individual, the same individual, it is going to be exhibit both the type of the symmetry during the growth phase. During the larval condition, it will be exhibit bilateral symmetry. The larva of echinodermates, they are exhibiting the bilateral symmetry. The adult individuals of echinodermates, they are exhibiting the radial symmetry. Adult ones, they are exhibiting radial symmetry. The larva ones, they are exhibiting the bilateral symmetry. Individually same, but during the different stage of the development, they are exhibiting the different symmetry. They are exhibiting different symmetry. So that's why it is called the biradial symmetry. So in echinodermates, it is common. The larva of the most of the echinodermates, they are exhibiting the bilateral symmetry, while the adult ones, they are exhibiting the radial symmetry. That is the radial symmetry. So based on the type of symmetry, the organisms are classified into the different groups. The polyphenols, most of the polyphenols, they are asymmetry. That's why these are kept in a separate phyla. The organisms, they are exhibiting the radial symmetry, and the blind sac body plan based upon the type of body wall, body layers. So these are classified into the phylum Ctenophora, phylum Coelenterata, phylum Echinodermata, the phylum Platyhelminthes, Aschelminthes. The bilateral symmetry is common as in case of the Platyhelminthes individual. The first time these group of individuals, they are exhibiting the bilateral symmetry. Likewise, the Aschelminthes individuals. So apart from these, even though we are classifying the organisms not only based upon the symmetry, even though based upon the metabolism also, the segmentation also. Segmentation. Or metabolism. What do you mean by this metabolism? What do you mean by this segmentation? The segmentation is nothing but serial repetition of body parts. Serial repetition of the body parts is referred as the segmentation. The body parts are repeated. Consistently, serially, they are going to be repeated where they are possessing the repeated body units, or maybe the body parts, or simply we can say the repeated body units. The repeated body units is called as segmentation. So whatever the repeated units are there, so these repeated body units are referred as metameres. These are referred as metameres. These are also called as the somites. These are also called as the proglottids. These are also referred as proglottids. Metameres or somites or proglottids. The repeated units, or maybe the serial repetition of the body parts, is nothing but it is like this. The segmentation we are going to observe, like this segmentation. So this is one segment. This is another segment. So here is another segment. Then here is abdominal wall. Abdominal wall also possessing the repeated serial units. Here it is like this. So these are referred as segments. Segments are these are also called as metamere. A single segment is called as metamere. The single segment is also called as a somite. The single segment is also referred as proglottids, but it is in different group of organisms. That is called segmentation. That is called the metabolism. So where the body of an organism is differentiated into different segments. So if you observe in arthropods or maybe in some of the other organisms where the body it helps in, the segmentation is helps in, body is divided into the head, thorax, abdomen, head, neck, thorax, abdomen, head and trunk and tail, like this. This is mainly because of the segmentation. This is because of metabolism. The metabolism is helps to divide the body into the different parts. So if some segments are also present in the head region. So these segments are called as cephalic segments or head segments. The segments are present in the thoracic region. These are referred as thoracic segments. The segments are present in the abdominal region. These are referred as abdominal segments. Likewise, this segmentation or maybe metabolism is playing a vital role. Differentiation of the body into the two different segments, two different parts. Internal, based upon the internal segmentation as well as the external segmentation. So you are going to observe the external segmentation in most of the arthropods. Where, so these are the external segmentation. On outside, if you observe these organisms, we are going to observe the different segments. External segmentation. Even though internal segmentation is also observed, where the body is metamerically segmented ones, as in case of annelidans, as in case of arthropods, as in case of annelidans, both the internal as well as external segmentation will be there. But as in case of arthropods, only external segmentation is there. Internal segmentation we cannot observe. Because of the presence of external as well as internal segmentation in annelidans, the type of metabolism is called as a metamerically segmented body. True metabolism. But the metabolism might be is the pseudo metabolism. Let us see. The first one. True metabolism. Second one. Pseudo metabolism. Pseudo metabolism is nothing but it is a false metabolism. Not true. In true metabolism, the first formed segment is oldest one and last formed segment and last formed segment will be youngest one. First formed segment will be oldest one and the last formed segment will be youngest one. The first formed segment is present towards the anterior surface of the body, otherwise it is present towards the posterior surface of the body. But these first formed segments are the oldest ones. These are old segments because these are first formed from the developing embryo. And these are becoming an old ones. And the last formed segments will be the youngest ones. So that is called as true metabolism. That is called as true metabolism. But as in case of pseudo metabolism, the true metabolism we are going to observe right from the Ascalminthis up to the even the in Ascalminthis there is no metabolism. The body is not exhibiting any metabolism. Metabolism we cannot observe. The segmentation is not there because the body is made up of muscles. The soft body. So that's why Ascalminthis individual they do not exhibiting any metabolism. But as in case of the annelidans, if you consider the first formed segment in annelidans, it is an oldest one and it is present towards the anterior surface of the body that helps in formation of the head region, that helps in formation of the head capsule, as in case of arthropoda. That is true metabolism. But even though we are going to observe the false metabolism, the pseudo metabolism in some group of organisms, the example for true metabolism is if you take Annelida, arthropoda, chordate individuals, so where they are exhibiting the true metabolism, here the false metabolism or pseudo metabolism. Example is here it is the platyhelminthes individuals. Platyhelminthes individuals. So where in pseudometabolism, first formed segment is youngest one, and last formed segment will be oldest one. This is reverse, exactly reverse of the true metabolism. Now, whatever the first segment is found in the platyhelminthes individuals, it is youngest one and it is present towards the anterior most part of the body. And whatever the last formed segment will be there, instead of it is youngest one, it becomes an oldest segment. So the last formed segment will be the oldest one, the first formed segment will be the youngest one, and the youngest segment is present towards the anterior surface of the body. So that is called as a pseudo metabolism, false metabolism. So if you observe the false metabolism, It is like this, where the separate head formations occurs, but whatever the first segments are there under the anterior surface of the body, these are becoming an youngest one. Youngest segments these are. But whatever the last form segment will be there, this last form segment will be the oldest one. Actually, the first form will be the oldest one. But here it is, last form segment will be the oldest one, first form segment will be the youngest one. That is called as pseudo-metabolism. But whatever the segmentation we are going to observe here, this segmentation, whatever the segmentation possessing the segments, so the segments of these plateal mantis are referred as the proglottids. These are referred as proglottids, not somites, not metameans. These are referred as proglottids because these proglottids may be of three different types. Here, whatever the proglottids are there, so these proglottids are immature proglottids, and whatever the middle portion of the body proglottids are there, so these are ones matured ones. These are mature proglottids, and whatever this posterior end of the body proglottids are there, so these proglottids are called the gravid proglottids. Immature proglottids, matured proglottids, and the gravid proglottids. The immature proglottids, they are not at all involving in any sexual or maybe asexual reproduction. They are not at all involving in any sexual or maybe the asexual reproduction, but these are youngest ones, present towards the anterior most part of the body. But whatever these mature segments are there, these mature segments or maybe proglottids, they are involving in a sexual reproduction by the formation of the gametes. By producing the gametes, they are involving in sexual reproduction. But whatever these gravid proglottids are there, so these gravid proglottids, by detaching from their parental body, and they are involving in asexual reproduction. Those are referred as the gravid proglottids. So this is about the pseudo-metabolism, where in this group of individuals we are going to observe first time there is a formation of the cephalic end, that is anterior end of the body. This anterior end or maybe cephalic end of the body is mainly made up of the fusion of the certain segments. So these segments are youngest segments. They are mainly possessing the certain sensory structures that helps in formation of separate head region. Separate head region. That is called cephalic end or maybe anterior end. So this is the false metabolism, pseudo-metabolism. But as in case of other individuals, other group of organisms, whatever the segments are there, so these segments are the true segments. So these two segments, they are exhibiting either internal segmentation or it is external segmentation, or in some individual, some group of individual, they are exhibiting both internal as well as external metabolism or segmentation. The metabolism is again divided into the external metabolism. Second one is internal metabolism. Third one is external as well as internal metabolism. External as well as internal metabolism. The external metabolism is common as in case of arthropoda individuals. So based upon the metabolism, we are classifying, based upon the type of metabolism, we are classifying the organisms into different groups. The organisms which are exhibiting only the external metabolism, these are kept under the phylum Arthropoda. They are kept under phylum Arthropoda, along by considering the symmetry. Apart from that, the coelomic cavity, body wall, organ grade of organization, tissue grade of organization, organ system grade of organization. By considering these basic characters, so we are classifying the organisms into different. So here, based upon the type of metabolism, we are again classifying organisms into different groups. Like external metabolism, the animals or maybe the individuals, they are exhibiting the external metabolism. These are kept in arthropods. If you take the crab, if you take the millipede, centipede, or it might be the prawn, or it might be the insects, the thousands of insects, lakhs together insects. So where the body of these insects is mainly classified into head, thorax, and abdomen. So this is mainly because of the effusion of the certain anterior most part of the body segments or maybe metameres to form one of the head capsule, which helps in formation of the head. And some of the metameres, they are helps in formation of the thoracic segments, that helps in formation of thorax. And some other metameres, they helps in formation of the abdominal segments, that is abdomen. So that is, we are going to observe only external segmentation here, external metabolism here. Externally, we are observing, we are differentiating the body into different parts. So like these segments, we are going to observe it externally in this group of organisms. Not internal. Internal segmentation you cannot observe in arthropods. But as in case of the human beings or mammals, internal segmentation is there, internal metabolism is there. This internal metabolism is mainly because of the presence of the certain vertebrae, apart from that muscles. And also the internal metabolism is mainly helps in the dividing the body into the two different cavity, that is a thoracic cavity from that of the abdominal cavity. This is the thoracic cavity. Here is the abdominal cavity. The thoracic cavity gets separated from the abdominal cavity due to the presence of the muscles, like the diaphragm. Diaphragm is mainly made up of muscles. Diaphragm is mainly made up of muscles. The diaphragm separates the thoracic cavity from the abdominal cavity. That is internal metabolism, internal segmentation, which is observed in mammals, like the human beings also. This is because of the presence of the vertebrates, or maybe it be the muscles. Then, the external as well as internal segmentation. The external and internal segmentation, both the type of segmentation is common as in case of the organisms belongs to the phylum Annelida. That's why the Annelidans are referred as the metabolically segmented ones. These are referred as metabolically segmented individuals. The body is metabolically segmented. The body is possessing the repeated body parts or maybe the body units. The repeated body parts are maybe the repeated, the linear, repeated, or maybe serial, repeated, the body units or maybe the body parts. That is helps in formation of external as well as internal metabolism. The external metabolism we are going to observe, even though internal metabolism also we are going to observe. External metabolism is mainly occurs due to the presence of annuli. The internal metabolism is occurs due to the presence of septa. So that's why, so the individuals belongs to the phylum Annelida. They are exhibiting both internal as well as external metabolism, external segmentation, internal segmentation also. So that's why each and every segment, they are possessing a part of the particular system. A part of the digestive system is present in a particular segment. A part of the excretory structures are present in the particular segment. A part of the circulatory structures are present in a particular segment. A part of the respiratory structures are present in the particular segment. That means each repeated units of the body, they are possessing all the different types of systems, a part of the systems. A part of the different systems, they are present in each and every segment of the body. So that's why the annelidans are referred as the metamerically segmented individuals. They are referred as metamerically segmented individuals. So if you observe each and every part of the body, so particular segment, every segment, if you observe, if this segment number 1, or might be the segment number 28, or might be the segment number 4, or maybe 6, or maybe 10. If you take any segment, this segment possessing not only a part of the digestive system, a part of the circulatory system, a part of the excretory system, a part of the respiratory system, all these systems, a part of the systems are present in a particular segment. That's why it is called as a metamerically segmented one. So that's why, so in this group of individuals, like the earthworm, like the earthworm individuals, where the body is gets fragmented into different fragments. Different fragments. The fragmentation type of sexual reproduction is common as the case of this earthworm individual, because mainly possessing the repeated body units in each and every segment of the body. So that's why such type of metamerically segments are referred as, they are referred as somites. They are referred as somites. That means the segments present in annelidans, so they are referred as somites. So that is external line, internal segmentation. Next, the cephalization. What do you mean by this cephalization? The cephalization is nothing but the concentration of the various sensory structures, or maybe the organs, towards the anterior end of the body, towards the anterior or cephalic end of the body. That helps in formation of separate head. That is called cephalization. The concentration of various sensory organs or maybe structures. The certain sensory structures or maybe the organs, they are present towards the anterior end or maybe towards the cephalic end of the body, that helps in formation of the separate head region. That is called cephalization. The formation of the separate head is first time is observed as in case of the organisms belongs to the phylum Platyhelminthes. These are the individuals, these are the first individuals who belongs to the phylum Platyhelminthes. They were exhibiting the cephalization. First time we are observing the formation of the separate head region in this group of organisms. So as I said, the head region, as in case of Platyhelminthes, as already I drawn, so where they are possessing the hooks as well as the suckers. Hooks as well as suckers. Apart from that, they are also possessing the numerous sensory cells which are tactile in nature. Such type of sensory cells are present towards the anterior end or maybe cephalic end of the body. This anterior end or maybe the cephalic end that intermodified into separate head region. So this head formation is occurs due to the fusion of certain group of segments. Certain segments, they are fused to form a separate head region which possessing the various sensory cells or maybe the sensory organs. That is called cephalization, which is commonly observed in Platyhelminthes individuals, where the hooks are there. Hooks are helps in the attachment to the host body. These hooks are gets attached to the host body through particular sensory cells. Apart from that, the surface of the head region also possessing the tactile receptors. They are possessing the tactile receptors. The suckers are present. These suckers are helps in the absorption of the nutrient from the host body. Because the most of the individuals belongs to the Platyhelminthes. These are endoparasitic ones, and few are ectoparasitic individuals. So because of their endoparasitic nature, they do not possessing a highly well developed digestive system. That's why the system level of organization we cannot observe in this group of individuals. So that's why these individuals are exhibiting organ grade of organization, not organ system grade, not tissue grade. All the different types of tissues, they are aggregated to form one of the organ. The particular organs are helps in formation of the body construction. So that's why these are exhibiting organ grade of organization because of the endoparasitic mode of nature. Apart from that, so these are organisms where they are commonly called as the flatworms. The name itself explains here. The platy means it is flat. Helminth means it is worms. It is flat worms. Where the body of these individuals, it is a dorsoventrally compressed ones. The body of this group of individuals, the body is dorsoventrally compressed. Dorsal and ventral surface from the both sides it gets compressed. That's why they do not possessing any cavity. So that's why these group of organisms are referred as coelomates. So later on I will explain regarding about the coelomic cavity. Coelom. In next class I will explain regarding about coelom. So that's why these are coelomates, but they are triploblastic organisms exhibiting bilateral symmetry, exhibiting organ grade of organization, pseudo metabolism, but they are possessing definite head region. They are possessing definite head region with certain sensory cells or maybe sensory structures. That is called cephalization. So the remaining portion I will continue in next class. Thank you.